Good morning, everyone, and thank you to the organisers for allowing me a chance to come and speak to you today on David Barker's commemorative meeting. I'm really sorry that I can't be here in person today. I have to be at a meeting in Spain. It's an EU Framework 7 meeting where we have a lot of the people from Brussels turning up, and I just have to be there. But my heart, or at least my digital heart, is in Southampton with you all today. So, embryos and David Barker. About 15 years ago, David and I met for the first time, and he introduced me to this whole subject of developmental programming, or fetal programming as it was called then. At the time, I was a more classical developmental biologist working within the biology department, interested in how blastocysts formed, how from the time of fertilization the cells decided what they were going to become, cell interactions, those kind of things. But at the end of the day, it was very much centered or centric around how the embryo developed. And meeting David for the first time gave me the opportunity to think more broadly. He said to me, you know, Tom, it's all very good studying how an embryo develops, but you've got to think also about what's happening outside the embryo, how that might be influencing how the embryo develops. And this, of course, opened up a whole new world for me. And through interactions with the Faculty of Medicine at the time, and I remember and appreciate the help from Eric Thomas and Mick Arthur at the time, he was able to arrange a postdoc to work in my lab for a year or so at that time, to allow us to start studying whether or not the very early embryo had a role in developmental programming. And that was Lydia Kwong, and that got us going in the subject area, and I've never regretted a moment of it since. So thank you, David, for that opportunity to work in what is, I feel, the most exciting area of biomedical science today. So on this second slide, what I've done is just to indicate the area in development that we think is very important in terms of developmental programming. That ring on the top left shows the early embryo phase, and it's right at the start of development, and there's a lot of development has to pursue after that before birth. So we're looking at a very, very early stage of development when we think the environment can influence the developmental program with lasting and enduring effects into adulthood of the next generation. So why would we feel that the early embryo is a key stage in developmental programming? As this picture here shows, the embryos just have a very small number of cells at this stage, immediately after fertilization. It represents a 3 or 4 day or 5 day period depending on species. And even at the end of it, when a blastocyst is formed, it's a very small number of cells. So one reason why embryos can be important in developmental programming is the fact that because there are small cells, any change or environmental effect on those cells is going to have a lasting effect because there are so few of them. All of the subsequent lineages that form are going to be affected from this early environmental interaction. Secondly, the embryo is totipotent at that stage. That is to say, it has the capacity to form all of the lineages subsequently, both the full repertoire of lineages that make the fetus and baby, but also the extra embryonic lineages, as we'll talk about in a minute. So that stage of development is one where the embryo can, if you like, interact and respond to the environment, affecting both the placental lineages as well as the embryonic lineages. Thirdly, there's a lot of epigenetics that goes on at this stage. Major changes in the coating of the chromatin, DNA methylation, histone modifications that can affect the developmental program and influence how changes may be propagated from that time onwards. And the way we think it, and this really originated from when we had the conversations with David, was perhaps the early embryo will assess its environment and actually make changes to how it will develop according to the information it receives from that environment to optimize its program development, to optimize phenotype after birth. this kind of concept of plasticity that's developed since. So over the years, since those initial interactions with David, we've used a whole range of different models. This slide here indicates the effect of ART, IVF technologies, assisted reproduction technologies, but also maternal sickness, maternal obesity, and in particular, as indicated at the bottom, the importance and effects of protein deficient diets, the low protein diet model that we've focused our work on. And I'm just going to summarize a few key points from that work today. So, embryo low protein diet. This is the concept of what happens if you feed a mother using our mouse models and earlier rat models, feeding a low protein diet just for the period of the pre-implantation embryo development, about 4 days. Well, and then moving back to normal control diet for the rest of gestation, and indeed normal diet for the offspring right the way through to adulthood. If we do those, if we make that simple treatment, we can see a number of long-term effects. As indicated on the slide, the adult cardiovascular system is dysfunctional. You have hypertension, you have poor arterial dilatation, and other cardiovascular phenotypes. We also have metabolic dysfunction, with ultimately the mice becoming high in adiposity, increased growth in later life. Thirdly, a very hyperactive abnormal behavior patterns. So across the whole spectrum of different systemic systems in the adult offspring, we see changes, all derived from that very brief period of protein undernutrition fed to their mothers when they were pre-implantation embryos. One of the other characteristics that we see from this model is that there is a stimulation of fetal growth, and indeed perinatal weight is stimulated from this dietary treatment. And indeed, that increase in weight is a predictor of future disease risk. If you do correlations between weight and future disease, then it's very positively correlated. So one of the things that's important in our work is to try and identify how does that increase in growth, how is that process initiated, because it is a true marker of future disease in the adult offspring. So on this slide, I've tried to just summarize using that embryo low protein diet, originated, I should say, from Alan Jackson's lab. What we think from a number of studies, what we think are the key steps in the process of developmental programming, the sequence of events. And if we look here, we see the first thing we can identify that change in diet changes the mother, changes the composition of nutrients within the serum, and that translates through to the composition of the uterine fluids. We see changes in the uterine fluid composition of particularly amino acids. And if we look then at the embryos, we find that the embryos have sensing mechanisms that can detect the quality of those nutrients present in the uterine fluid. And there are signaling pathways, particularly the mTOR pathway, that changes in its signaling activity as a consequence of that changed composition in the uterine fluids. That's a way for the embryo to sense and know the quality of the nutrients that it's bathing prior to implantation. It's at that stage, in the blastocyst stage, that we think the embryo becomes reprogrammed to change its pattern of development, ultimately leading to those disease conditions that I spoke about earlier. So if, for example, we take a blastocyst from a low protein mother and put that blastocyst into a mother that's been fed just a normal protein diet, so the changes that occur in programming from the low protein will persist even in a normal mother. So that's an important point. So the induction of programming occurs at that stage. The responses by the embryo are those then that, in broad terms, lead to compensation, attempts by the embryo, which has its own agenda in life, if you like, attempts by the embryo to increase the nutrient supply from the mother, to make it competitively fit, to allow it to pass on its genes, but has this drawback that ultimately will link with disease in later life. And we've seen many of these different compensatory processes, but the key fact, and this is why the embryo is such an important stage to study these things, it affects the extra embryonic lineages. So that little diagram that we see on the bottom left of the screen, the outer cells of the embryo are extra embryonic lineages, the trophectoderm that goes on to form the placenta, and then the purple layer within the inner cell mass above the blastocele, the primitive endoderm, also goes on to form, in this case, the yolk sac placenta. So those two lineages, the trophectoderm and the primitive endoderm, will not form part of the fetus, but respond to the early nutritional challenge to change the functioning of the placenta, both the chorioallantoic placenta from the trophectoderm and the yolk sac placenta from the primitive endoderm. And we'll look at some of those changes now very briefly. So on this slide we see a series, or summarized a series of different changes mediated in these extra embryonic lineages. The trophectoderm and the primitive endoderm have a stimulation of endocytosis, because if you can't get the availability of nutrients directly from the mother, there's the possibility to increase the uptake of those nutrients quantitatively through endocytosis. And there are cell biological systems, particularly involving Rho-A GTPase, that allow that process to occur. There's also proliferative stimulation of these lineages. We see that the number of cells forming the extra embryonic lineages is increased in the embryo as a result of that low protein diet. So there's a change, if you like, in the morphogenesis of the embryo, a greater focus on developing a more efficient extra embryonic lineages to try and get more nutrients. And then the invasiveness of the trophectoderm at implantation increases. The motility and capacity to invade the uterine endometrium is stimulated. Again, another compensation. So to wind this up, we can see that from the very earliest stages of development, the capacity for the embryo to sense the quality of its environment. There are signaling systems to interpret that interaction, and a series of compensatory responses that ultimately will promote growth. But it's that growth promotion that will be a forerunner, a marker for later, for disease in later life. So we can see a continuum, if you like, between the changes occurring within the very early embryo before implantation and indeed poor health in adulthood. We're now developing in vitro models where we can use embryos without a diet, but derived from normal mice, and putting them in the environment that we think that the maternal amino acid and insulin composition may be inducing this programming from the uterine fluids. And when we generate those in vitro environments, we indeed are inducing programming similar to what we find in vivo. So it's giving us greater confidence that we've understood the pathway of mechanisms from the embryo period onwards. We're also looking at the epigenetic changes on the key genes, key transcription factors that affect early development through programming, and particularly those within the extra embryonic lineages. And lastly, we're now making use of embryonic stem cell lines derived from dietary programmed embryos to allow us to study the mechanisms in further detail without having to use animals, which is a great advance. So I've told you there about some key factors in the story from the low protein diet model, but just to also say, from in vitro culture and assisted reproductive technologies, the environment of the embryo is different from in vivo, and we see there a series of similar related changes to those that I've talked about from the dietary program. Changes in the way the blastocyst morphogenesis program occurs, in the cell allocation of lineages, and ultimately changes in cardiovascular and metabolic behaviour in the adult. And indeed, some similar phenotypes are now found in the human from IVF children. A completely different model is what happens to a mother when she becomes sick at the very onset of development. And here we've developed a model to mimic a bacterial infection using an endotoxin, and that too has programming consequences. Here, a different type of programming where sickness raises the maternal cytokines. That's detected by the very early embryo, we think through toll-like receptors, and results in a change in the developmental program. But here, it's not the cardiovascular system that changes, but it's the immune system, the innate immune system of the offspring. So it suggests that, again, whilst the embryo is sensitive to environmental change, different types of environments can induce distinct long-term effects. So that's all I'll say. My heart is with you today. David's had a fantastic influence on my career. I'm so grateful to have met him and worked with him. And a big thank you to him and all the people that have worked in my lab, and indeed various collaborators here at Southampton that have made my time here so exciting. And have a great day. Many thanks for your attention.